So, good evening, everyone. Yesterday we had just the introduction of anatomy of flowering plant part 2, where we discussed that plant tissue can be divided into three areas depending on their position. This is epidermis, then ground tissue, and the vascular bundle. So I'm just quickly scrolling that these are the things, these are their position. We learned about epidermal tissue, what are the components of epidermis that is observed in plant, what are stomata, what are their significance, what will happen if guard cell is not present in the stomata. We also studied about epidermal appendages too. Apart from that, we studied about ground tissue. I told you depending on plant and depending on plant part, that is root or stem, the ground tissues will be different. Common basic structures will be there like cortex, but the area of cortex, how it is arranged, it is different. Then we talked about vascular tissue, that is xylem and phloem. Depending on plant and that is plant in terms of monocot or dicot, also body part, that is root or stem or leaf. The vascular bundle, that is combination of xylem and phloem, can have various types. We discussed yesterday about open and closed type of vascular bundle, as well as conjoint and concentric vascular bundle, that is also called radial type. So today we will discuss mainly about the anatomy of dicot and monocot plants. Okay. So we will start our discussion with dicot root or dicotyledonous root. The four main parts of dicot root are epidermis, that is the outermost part. See, I am leveling them. It is having here pink colored, but don't worry, in plants they are not pink colored, actually they are colorless. And in case of dicot root, these tissues are generally barrel shaped. As they are present in the root, the layer of cuticle will be absent, because in presence of cuticle they cannot absorb water. You can see that in epidermis region there are presence of root hairs, and all these root hairs are unicellular in their nature. As this part is deleted in board examination of CBSE, you don't have to as such practice the diagram, but in Gujarat board it is there. So a favorite question of examiner is draw and describe this part. So if they ask you, kindly practice in advance, because it should not have a beautiful diagram but a neat and clean diagram. So I suggest in our next test we can put this. And for CBSE students, any question can come in need with diagram, and they can tell you to mark the labeled part or write your function or anything related to diagram can come. So you don't feel happy that okay, this diagram is gone. You also need to remember the diagram, and for remembering the best way is to draw it. So both ways we don't have much option left. Okay. So after epidermis region there is cortex. You can see the majority part of the root is cortex. Okay. Then there is endodermis. You can see this layer of endodermis which is encircling the vascular bundle. So let us know about each of them in detail. Okay. So the outermost part is epidermis. It is made up of mainly parenchyma. and it has unicellular root here. Then is cortex. It is also made up of parenchyma cell, and generally they are thin walled with intercellular spaces. Why thin walled? Because root cells need to be living and active. Why? It may not be active in terms of division, but they need to have a activeness. Why? Because they need to go for the transmission of water. That's it. Okay. So post cortex there is endodermis, and in this innermost layer which is present innermost to cortex, which is called endodermis, there will be again presence of barrel shaped cell. Can you tell me why barrel shaped cells are present in epidermis, endodermis? What is the significance of this barrel shaped cell? Do you know what is barrel? Beak. Why these parts are having barrel shaped cell? हाँ, इसमें पानी स्टोर होता नहीं. एपिडर्मिस इज फॉर प्रोटेक्शन. They actually can have minimum intercellular space, and whenever you want to cover something, I told you while teaching epithelial tissue that it should not have much intercellular space, otherwise the purpose of protection will not be done. अगर आपकी दीवार में हद से ज्यादा छेद रहे तो वो दीवार किस काम है ना? So same way, that's why the most important layers are barrel shaped. Okay. Now, if you see these epidermis cells closely, you better look into the diagram portrayed in the same page on which I am moving my pointer. You can see that they have deposition of suberin. So what is this suberin? Suberin are water impermeable waxy material, and these material are stored in the form of strips called Casparian strips. They are stored in the form of Casparian strip. Now again there is a doubt. इसके अंदर vascular bundle है और एंडोडर्मिस के बाहर अगर वाटर इम्परमिएबल लेयर है तो ये ट्रांसपोर्ट कैसे करेंगे? बट फिर भी है. व्हाट इज द रीजन? टिश्यू डैमेज हो गया. कौन सा टिश्यू? जाइलेम से टिश्यू से इतने फोर्सफुली पानी नहीं निकलता है जब पाइप फट जाती है जितना फोर्सफुली पानी जाता है. बट तुम्हारा आंसर सही है. इट इज प्रिवेंटिंग वाटर लॉस. इट विल एंश्योर कि ड्यूरिंग ट्रांसपोर्टेशन इट शुड गो थ्रू द वास्कुलर बंडल only. Sidewise transportation doesn't take place. That's why it has those impermeable layers around it. Next is steel. What are these steel? Steel is the central part which includes vascular bundle. Many a time students get confused about vascular bundle and steel. Vascular bundle is a part of steel which has xylem and phloem. But xylem and phloem cannot be suspended in air. They require some kind of support. So steel will include pericycle, just below endodermis, a few layers of thick walled parenchyma is there. They are forming the pericycle. What is the role of pericycle? The lateral roots and vascular cambium, which is part of secondary growth, will come from this steel part, which is pericycle. So pericycle is a part from which the lateral roots will come, as well as secondary growth will initiate. Next is pith. What is pith? You can see these orange colored structures are xylem phloem. Okay. In between that center, there are cells. Can you see my pointers clearly? So that part is known as pith. There are some parenchymal layer which is present between xylem and phloem, that is called conjunctive tissues, and vascular bundle will include xylem and phloem patches. So you can see the xylem and phloem are arranged in which manner? Are they conjoint or concentric? Concentric. Concentric. Okay. And as it is a dicot root, do you think it will be having open vascular bundle or closed vascular bundle? It will be having a open vascular bundle. What I said to you, the vascular bundle in which cambium is not present. Whenever I am mentioning about secondary growth, that means cambium will be present. So cambium will exist between xylem and phloem. So they will not be close to each other, no? So that's why open. In closed vascular bundle, the xylem and phloem will be very, very close to each other. Okay. So this is the structure of monocot root. It will include epidermis, cortex, endodermis, pericycle, vascular bundle, and pith. So it has more than 6 vascular xylem bundle. That's why called polyarch, and the pith is comparatively large. So as soon as there is completion of this part that we are studying today, for our next lecture on Monday, you people will have a homework. Okay, what is this homework? The homework is you need to take a full scale white page, draw the diagram of root of dicot, root of monocot side by side, and write down the layers. It will help you to remember the similarities and dissimilarities. For example, the epidermis of dicot root, epidermis of monocot root, they are similar. No difference is there. Cortex of this and that. But when pith will come, the pith of monocot will be larger than the dicot. Okay, so once and all you are done with this exercise, next time onwards you don't have to study from book or need to refer this video. You have ready reference in your hand through which you can directly discuss about the difference and comparison. And it will not allow you to get confused also. Okay. So as the why pith is can afford to be learned, because in monocot there is no cambium and it doesn't go for any kind of secondary growth. Now we will talk about dicot stem. So before we talk about dicot stem, I wish all of you to have a look into your textbook and let me know if you have any doubt in monocot root or dicot root. So as you people confirm that, as you people confirmed in root, you don't have any doubt. So we will move toward dicot stem. So basically in stem, cortex region is much larger. So what are the differences? Kindly note down. In epidermis, it has thin layer of cuticle. It has trichome, which is multicellular. It has stomata. Stomata are strongly absent in root. So after epidermis, there is cortex. Here, this layer is subdivided into hypodermis, cortex layer, and endodermis. Okay, so you can see that cortex layer is quite large over here. You can see here epidermis. Below that, there are parenchyma layer, then parenchyma layer, and finally there is endodermis. Kindly refer the diagram. So let us know about each of them one by one. Cortex can be divided into hypodermis, Hypodermis is called hypo because it is present just below epidermis. It is actually responsible for providing mechanical strength to the young stem. When the stem grows and there is formation of woody structure that we are going to study in secondary growth, hypodermis does not exist. क्योंकि सब कुछ अंदर से ग्रो होके क्रश होके then there will be formation of non living structure called bark जो हम देखते हैं थर्ड जो होता है प्लांट. Okay, but in young plant they require some kind of mechanical support which can be provided by parenchyma. Why not sclerenchyma? Because they require to be flexible and agile. It cannot be dead and dry and tough. That's why. So below hypodermis there is thin walled parenchyma cell and they have lot many intercellular space. This region is called cortical region. Then there will be endodermis. Cells are very rich in starch grain over here. That's why it is called starch sheet. Because whatever food is produced in leaf, it is coming from leaf to the stem region, and this is a very good storehouse of the food. If you look into the stellar region of dicot stem, you can see in this case also pericycle is present. So pericycle was present in root, it is present here also. But here a few differences are there. First of all, the vascular bundle here will be conjoined. So xylem and phloem will lie together, not side by side. Concentric में क्या होता है? Xylem और phloem एक दूसरे के बाजू बाजू है. In this case, they will be present together. That means if here is xylem, just below that there will be. They will not be locating side by side. Okay. So that's why between two vascular bundle, there will be presence of cells. जैसे आप देख सकते हो, I will point it out with different pointers. Here is one and here is one. So in between these two, you can see a layer of cell green in color is present, and that is known as medullary rays. One vascular bundle is here. You can see the pointer of biology. Other vascular bundle is here. They are pink in color, circular in shape. In between them, there are green cells, no? So they are called medullary rays, which are located below the two vascular bundles. Okay, and the pericycle present inside endodermis, just above the phloem. They are semi-lunar patch. Semi-lunar patch means half moon shaped patches. Okay, and they are made up of sclerenchyma. Because vascular bundle need to be protected, and this sclerenchyma layer here present is for protection. So vascular bundle conjoined and open because cambium is present in between. Protoxylem is endark. I told you already about endark and exarch. So pith, it is the central part. It is quite round. Always remember, you can see two diagrams here, 1 and 2. This is the cross section, first one, and the second one is जैसे केक मिलता है ना दुकान में, cross section is like a cake, but the piece you get is like this diagram. So वो circular होता है, उसको हमने triangular pieces में काटे हैं. So you can see the pith region is present here and it is quite wide, okay, and made up of parenchyma cells. So that is all about steel and finally structure of dicot stem. Let's have a look of monocot stem. It is quite different. By the look you can see, even if sometimes I feel that these vascular bundles are looking like a person, no? Two eyes, nose and mouth. But this is the way why I always mention this funny thing. You can always remember these. You will never get confused. Okay. So here it will have epidermis, which is quite similar. But the hypodermis are made up of sclerenchyma, not parenchyma. Why? Because monocot stems and monocot plants are not that woody. They are generally grass-like. That's why they have sclerenchyma. Okay. Yes, beta. होगा, but उसकी टफ मतलब have you ever tried ग्रास को कभी खींचने की कोशिश की है? हाथ में लगता है. So that hardness, that tensile strength is provided by this sclerenchyma, which is required because they don't have secondary growth. That is all they have. In case of dicot, those young saplings one day will become tree. So they will be protected because they will have maximum part dead. But in monocot, they will never become tree. So they need to protect themselves. No? That's why it is having sclerenchyma diameter. So these, why I say the things I am saying, no, they are not mentioned in any books. These are the tips which you should remember to remember the structure. Okay. So let's see that hypodermis is made up of sclerenchyma. In their case, the cortical region and the other regions are not prominent because in their case, the pericycle and the endodermis is not at all visible. Why? Vascular bundles are scattered. Can you see here how many vascular bundles are there in this figure? 4. So they are scattered. It's not the situation. In other cases, what you have seen, they are having tendency to locate at the center of the cross section. Here it is not the case. They are scattered. So these scattered vascular bundles, it's not possible then they have to have an endodermis around them, pericycle around them. That's why they don't have endodermis and pericycle. So what they have conjoined closed type of vascular bundle. Generally, the peripheral vascular bundles are smaller, and the one present at the center they are smaller, and the one present at the center are bigger. Now, each bundle. Then what about the protection of vascular bundle? How that is possible? वहाँ पे who was there for protection of vascular? Pericycle, and then there was endodermis. Here it's not there. So can we allow these vascular bundles to grow without protection? So in many plants, they have a specific sheath made up of sclerenchyma, which is known as bundle sheath. Instead of giving all vascular bundle protection at one time, they are giving protection towards particular vascular bundle. Okay. So phloem parenchyma is absent. Water containing cavities are present between the vascular bundle. Water containing cavities are possible because they don't have cambium. They don't go for secondary growth. Enough space they have. Okay. So let's move ahead. Now we will talk about leaf. I want you people to go and see leaves that you find around. It will take 5-10 minutes. While you are coming out of your house, you can look into plants, or even at your tuition center, leaves nearby, any leaf is there. Leaves are of two types. One is called isobilateral. It is called. So first here, as it is mentioned dorsiventral, I am mentioning dorsiventral. So what is dorsiventral leaf? Both sides are different. So if you look into them, The number of stomata present in the upper side is less, lower side is more. This is a natural way to decide that less evaporation is possible. If the upper side is more exposed to the sunlight. Apart from that, if you look into this type of leaf, you will be clearly see that upper surface is dark green in color and lower surface is comparatively light. So in such type of leaf, as stomata is less frequent or in some cases absent in the upper side, their anatomy, if you take a cross section of leaf, taking cross section of leaf is very difficult because they are very thin. But if you are able to take, you see difference in anatomy in the upper side and the lower side. So that is in case of dorsiventral leaf. Whereas in case of isobilateral leaf, the anatomy is quite similar. So let's have a look into the anatomy of dorsiventral leaf. In this case, two epidermis will be there. Why? Because it has two surface open to the atmosphere. है ना? Root या stem में क्या होता है? Only one surface. Because it is cylindrical. So only one surface is open. From inside it is not hollow, isn't it? Yes. So, but in case of leaf, if you look, the upper surface and the lower surface both are open. So it will have two epidermis. Okay. If you look into dicot leaf, the upper surface or adaxial and lower surface or abaxial surface, both surface have epidermis. It has cuticle. Generally, stomata are present more in adaxial, abaxial surface, that is at the lower surface. So, yes, beta. तभी तो अच्छा है ना. Too much transpiration will not be there. Sunlight is required for transpiration. So that means if you are keeping one plate full of water during night time, it will not evaporate. What are the factors control rate of evaporation? Temperature affects. It will be slow. Can you guarantee it will not be there? Try and put a plate full of water in a dark room. अपनी जो बाथरूम है ना, जहाँ सनलाइट ज्यादा नहीं आती है, वहां रख के देखो. बाहर रखोगे तो फास्ट होगा, बट वहां पे भी होगा तो सही ना बेटा. So let's talk about mesophyll tissue. So tissue present between two epidermis is called mesophyll tissue. Other than vascular bundle, obviously, it is made up of parenchyma. Two types of parenchyma are present. Okay, so what are these two types? If you look here, these cucumber shaped parenchyma are present. Can you see? They are called palisade parenchyma. Okay, and here these spherical structures or almost spherical structures are there. They are called spongy parenchyma. Can you tell me why this difference is required in dorsiventral leaf? Q. Because beta, if you see, palisade parenchyma are very tightly packed. Whereas spongy parenchyma, it's called spongy because it has intercellular space. So when you are absorbing air via stomata, it requires some space for that air to move. And in upper surface, anyway, stomata is not there. So why to keep intercellular space over there? And this shape of parenchyma will allow compact packing. And more the parenchyma, don't forget the main function of leaf is to go for photosynthesis. It needs to take care about the food of the whole plant. So that's why in the upper surface it has palisade parenchyma, which will be helpful for production of food. Understood? So let's move ahead and look into the type of vascular bundle. So in vascular bundle, you can see here, it has veins and midribs. The leaves are having veins and midribs. So vascular bundle is located at those regions only. Okay. So vascular bundles are surrounded by a bundle sheath cell, which is giving them individual protection, because as you can see that leaves are having veins and midribs everywhere. So they cannot be present at one part center like stem. So they are scattered, so they require individual protection. Generally, the dicot leaf is dorsiventral and the monocot leaf is isobilateral. You can see the structure of isobilateral leaf. It is quite simple. No such difference of palisade and spongy parenchyma is there. Stomata are quietly present in both the surfaces. Mesocephyll is not differentiated, but they have a specialized structure which is called bulliform cell. You must have noticed that the monocot leaves are quite long and they can bend easily. They don't break. So these bulliform cells are acting as a hinge. You know, doors there are hinge. आप पुराने जमाने के दुकानों में जाके देखो, जब शटर सिस्टम नहीं था, उनके पास दरवाजे होते थे जिनमें हिंज होते थे. तो ऐसा बड़ा दरवाजा, उसको फोल्ड कर करके ऐसे खोलते थे. देखा आपने? आजकल तो मतलब मेरे बचपन में मैंने ऐसे दरवाजे देखे जिसमें दो ऐसे होते थे, मतलब दो होते थे, ऐसे खोलते थे. जैसे आजकल तो दरवाजा एक मतलब किसी को पता भी नहीं है कि वैसे दरवाजे होते थे कभी भी. इस जमाने पुराने घर में होते हैं. अभी दरवाजा एक ही इसका होता है ना. So these bulliform cells are present in monocot because their leaves are very long. They require some kind of folding. ऐसे सेम तलवार के जैसे तो नहीं रख सकते ना. They can be folded due to presence of these bulliform cells. But they cannot fold, na beta. But they can fold up to a great extent. आप ग्रास को ऐसे फोल्ड करो ना, वो टूटता नहीं है. फिर पूरे लीफ फ्लेक्सिबल होता है. They don't bend at certain points, no beta. So in our next lecture we are going to discuss secondary growth. Today I am not discussing it here because we will be requiring clear idea about anatomy of the stem and root to know about secondary growth. Second thing I wish to mention here, the secondary growth of dicot stem is well described here. More dicot root secondary growth is there, but it's not well described. So only the things that is exceptional mention. But I request all of you, whenever you are studying, you should study both of them separately. Keep your notes prepared, both of them separately. जैसे textbook में सिर्फ exceptional part बोल के छोड़ दिया है, don't do it like that. Okay. So we will have a look into the video we saw last day. Give me some time to share the screen. So let's continue our video from the part we stopped yesterday. Kindly have a look into them, and the portion of secondary growth we will continue later on.
ये 10 गुना बेहतर स्टेन रिमूवल एंड 2 months लॉन्ग लास्टिंग शाइन. अर्बन कंपनी प्रोफेशनल क्लीनिंग सर्विस.
इसीलिए
consists of parenchymatous cells found in the cortex.
आइए 10 गुना बेहतर स्टेन रिमूवल एंड 2 months long lasting shine अर्बन कंपनी
cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells are collectively called stomatal apparatus. The epidermal tissue has epidermal appendages such as hair and trichomes. The root hairs are unicellular elongations of the epidermal cells, and they help absorb water and minerals from the soil. The epidermal hairs on the stem are known as trichomes. They may be branched, unbranched, soft or stiff, and may also be secretory in function. Besides this, trichomes prevent loss of water due to transpiration. The second type of tissue system is the ground tissue system. The ground tissue in primary stems and roots consists of parenchymatous cells found in the cortex, pericycle, pith, and medullary rays. In the leaves, ground tissue is composed of thin-walled chloroplast-containing cells, collectively called mesophyll. Let's now study the third type of tissue system, the vascular tissue system. It is composed of complex tissues, namely xylem and phloem, which together form vascular bundles. In the vascular bundles of dicotyledonous plants, cambium is present between the phloem and xylem. Due to the presence of cambium, such vascular bundles can form secondary xylem and phloem tissues and are known as open vascular bundles. On the other hand, in monocotyledonous plants, vascular bundles do not contain cambium. They are unable to form secondary xylem and phloem tissues and are thus called closed vascular bundles. There are two types of vascular bundle arrangements, radial and conjoint. The radial arrangement is generally found in roots, where the xylem and phloem in a vascular bundle are arranged in an alternate manner at different radii. In the conjoint arrangement found in stem and leaves, the xylem and phloem are situated at the same radius of the vascular bundles. The conjoint vascular bundles usually have the phloem located only on the outer side of the xylem. In the conjoint open arrangement, the cambium is present between the xylem and phloem, while in the conjoint closed arrangement, the cambium is absent. The plant body has an intricate arrangement of various tissue systems that aid its growth and development. The root is one of the key organs that helps distinguish between a dicot and a monocot plant. Let's observe the transverse section of the matured zone of roots to understand their internal structure. We'll begin by studying the anatomy of a dicot root. The outermost single-layered tissue of the dicot root is called the epidermis. On the epidermis, you will also find some extended epidermal cells, which are unicellular root hairs. The epidermis is followed by cortex, which is formed of several layers of thin-walled parenchymatous cells. These cells have intercellular spaces. The innermost layer of the cortex comprises barrel-shaped cells without any intercellular spaces. This layer is known as the endodermis. The tangential and radial walls of the endodermal cells have deposits of a water-impermeable material or suberin, which is also called Casparian strips. Suberin is a waxy, waterproof substance present in the cell walls of the cork tissue in plants. Going back to the transverse section of the dicot root, after the endodermis, a few layers of thick-walled parenchymatous cells are present in the tissue. This is known as the pericycle. Secondary growth, such as initiation of lateral roots and vascular cambium, takes place in the cells of the pericycle. Notice that the pith in the tissue of the dicot root is extremely small and undeveloped. Typically, there are 2 to 4 xylem and phloem patches in a dicot root. In the later stages, a cambium ring gets formed between the xylem and the phloem. Some radially arranged parenchymatous cells are found between the vascular bundles. These cells form the conjunctive tissue, which is specialized for storage of water. The tissues present inside the endodermis, namely the pericycle, vascular bundles, and pith, are collectively known as stele. Let's now take a look at how the monocot root is organized internally. In many aspects, it is similar to the dicot root. On comparing the two roots, we find the monocot root also consists of epidermis, cortex, endodermis, pericycle, vascular bundle, and pith. However, there are some differences. The monocot root can have many xylem bundles, whereas the dicot root does not have quite as many. Moreover, in a monocot root, the pith is well developed. Although dicot and monocot roots have some structural similarities, they differ in the number of xylem bundles and size of the pith and cortex. Moreover, secondary growth takes place only in the dicot root. In other words, these roots thicken over time. A stem is the main axis or stalk of a plant, and it develops from the plumule of a germinating seed. Let's examine the internal structure of a stem in dicot and monocot plants. Here is the transverse section of a dicot stem. The regions of tissues in this stem include epidermis, cortex, and stele. The outermost layer of the stem, or epidermis, consists of a layer of closely packed cells and is covered with a thin waterproof layer called the cuticle. The epidermis also contains stomata and hair-like outgrowths known as trichomes. The next region of a dicot stem is the cortex. It lies between the epidermis and the pericycle and consists of multiple layers of cells. The main function of this region is storage of food material. The cortex comprises 3 subzones, namely the hypodermis, the cortical layers, and the endodermis. The hypodermis is the outermost zone of the cortex, which consists of a few layers of cholenchymatous cells. These cells strengthen the young stem of a plant. Below the hypodermis are cortical layers of round and thin-walled parenchymatous cells with intercellular spaces. The third or the innermost zone of the cortex is known as the endodermis. It is a single layer of tightly packed rectangular cells which are rich in starch grains. Therefore, this layer is also known as the starch sheath. Let's now examine the composition of the stele, which forms the last region of a dicot stem. This region consists of the pericycle, the vascular bundles, and the pith. The pericycle is composed of sclerenchymatous cells, and it lies on the inner side of the endodermis above the phloem cells. These cells act as a strengthening material. The vascular bundles are located in a ring on the inside of the pericycle. This ring arrangement is a distinctive feature of the dicot stem. There are layers of radially arranged parenchymatous cells in between the vascular bundles, which constitute the medullary ray. Each vascular bundle in a dicot stem is conjoint, open, and consists of endarc protoxylem. The central region of the stem is filled with pith. It is composed of a large number of rounded parenchymatous cells with many intercellular spaces. Now that you've learnt about the dicot stem, let's study the monocot stem. The tissues of dicot and monocot stems are quite similar. However, there are some differences as well. In the monocot stem, the hypodermis is composed of sclerenchymatous cells, while in a dicot stem, it is composed of cholenchymatous cells. The monocot stem has scattered vascular bundles, and their arrangement is conjoint and closed. Moreover, water-containing cavities are present within the vascular bundles. On the other hand, in a dicot stem, the vascular bundles are arranged in a ring as conjoint and open. In a monocot stem, each vascular bundle is surrounded by a sclerenchymatous bundle sheath and a parenchymatous ground tissue. Moreover, the vascular bundles on the periphery are smaller compared to the vascular bundles in the center. Therefore, the essential differences between a monocot and dicot stem lies in the arrangement of the epidermis, ground tissue, and vascular tissue. Leaves are the most visible organs of a plant. They perform many crucial functions such as photosynthesis and transpiration, in which their internal structure plays an important role. A dicot leaf is also known as dorsiventral, as its upper and lower sides are different in structure. On observing the transverse section of a dicot leaf, we find that it has three main parts: epidermis, mesophyll, and the vascular system. The epidermis covers the upper and lower surfaces of the leaf and is coated with the cuticle. The epidermis covering the upper surface of leaf is called the adaxial epidermis, while that covering the lower surface is called the abaxial epidermis. Typically, the abaxial epidermis has more stomata compared to the adaxial epidermis, which sometimes may not have any stomata at all. The next part of the leaf is the mesophyll, which is located between the upper and the lower epidermis. It is also called the green tissue of the leaf as it contains chloroplasts. These cells have fewer intersections. The size of the vascular bundles depends on the size of the vein. In dicot plants, the leaves have reticulate venation, in which the veins are of different sizes. Let's take a look at a monocot leaf now. It is also known as an isobilateral leaf, that of a dicot leaf. However, there are also some key differences. Unlike a dicot leaf, where stomata are found in greater numbers in the abaxial epidermis, in a monocot leaf, stomata are present on both surfaces of the epidermis. Moreover, the mesophyll cells are in the leaves of monocots such as grasses. Veins and cells of the upper epidermis modify themselves as large, bubble-shaped, colorless cells, also known as bulliform cells. On absorption of water, bulliform cells become turgid and expose the leaf's surface. Conversely, in water stress situations, there is loss of turgor pressure and bulliform cells become flaccid. As a result, the leaves curl inwards to minimize water loss. Hence, a lot of activity goes on in the internal structure of a leaf. Since major food production takes place in leaves, they are aptly known as the plant's powerhouse of energy.
So with this we are completing today's lecture. We will study secondary growth in the next lecture. Bye to all of you.